Welcome back, friends. Welcome to another video tutorial from Shomu's Biology. We've been talking about all the human physiology lectures, and this is going to be a series of videos where we talk about the nervous system. And in the nervous system, we'll contain 5 different video tutorials. We'll start with the nervous system anatomy, that is the neuron anatomy. Then we'll talk about the membrane potential, action potential, ion channels that helps to maintain the membrane potential, and we'll also see the synaptic transmission. So let's talk about the neuron anatomy in this video. The structure of the neurons reflects their function. One part of the cell of the neuron, if you look at here closely, if I tell you to break down neuron in 2 essential parts, you'll see one part which is the part of the cell that receives incoming signal, whereas another part generates outgoing signal. The part that receives incoming signal is dendrons, and they're connected to a cell body, while the part that transmits that signal, outgoing signals, is known as the axon. The goals for our learning is to understand the basic anatomical feature of neuron and the function of each anatomical region, and also to understand the information flow in neurons and the structural basis for communication between the neurons. And not only will see the communication between 2 adjacent neurons, but we will also see the interaction of a neuron with muscle and the interaction of neuron with glands. Okay, so stay tuned and watch the video to learn these topics. The first thing I should tell is the important feature of neurons, that the neurons can communicate. Because of their unique anatomical design and because they are excitable and suppressible, okay, they can communicate. The communication of neurons that we know is based on 3 different types. Just let me pick a color. Let's take a black color here. Neurons communicate with each other, which is a neuron-neuron interaction. It communicates with muscle. We call it neuromuscular junction. And the third one, neurons will also control the release of different proteins and secretory hormones from glands. Now the interaction between the neurons, if it's interacting with each other, is based on the dendron and axon interactions. Because as I told you, this is the cell body and the section of a bit which is receiving the signal, and another one is axon. So axon of one neuron interacts with dendron of the other neuron, and this is the way they interact. In case of muscle, if you look at here in this picture, you see dendron of one neuron interacts with the axon of the other. While if you look at the interaction of neuron and a muscle, you see there is a space between neural and muscle where the neuron interacts with it. Similarly in case of glands as well. Neurons have 3 characteristic structural features. They come in many different shapes and sizes. In this module we will examine the most common central nervous system neurons, okay, and the multipolar neuron. If we look at here, the typical structure of a neuron is consisting of 3 different parts. A cell body. This is the cell body of a neuron, where they contain the nucleus and all the other cellular components inside, the cytosolic sections. The second one is a receptive portion. Receptive portion means through which they will receive the signal, and that is nothing but the dendrites, okay, which are cytosolic extension of the cell body. And the third one is a transmitting portion, that is nothing but the axon, which is a thin rope-like structure coming out of the cell body. In the most neurons, the cell body or soma is located centrally, okay. Multiple similar processes called dendrites, as we see here, they come from this soma, forming a structure resemblance of a branch of a tree. That's why if you look at neurons, you'll also see this kind of branching and debranching, all the structures just like a tree. and the thin rope-like structure going out from it, holding it together. This is the characteristic structure of a neuron. The neuron structure is related to its function, and I told you earlier as well. These structures are very important in perspective of how the neuron will properly function. For example, if you look at the features of the neuron and the functions, for example, dendrites, cell body, and axon, those are the three things that we have. Among them, dendrites, these are the receptive part, okay? And the branch dendrites receive signals coming from the other cells and send them, so they will receive signal, and it will send them and channel them through the cell body and through ultimately to the axon. Okay, so they actually gather the incoming signals. The second one, which is a cell body is required for maintaining all the structures, right? This is a receptive as well as integrative region of the neuron. It is the main nutritional source and metabolic location of the neuron, because the other structures are very thin and they are the extensions of cytosol, while the ultimate process and job should be done inside the cell body. Although it also receives signal from the other cells and sends them towards the axon. So the work of cell body and dendrite is similarly working together to gather the signal and transmit it towards the axon. And the third one, the axon, the thin rope-like structure coming out of the cell body. This is the transmitting or conductive region of the neuron. The axon generates the action potential. It's known as the action potential. So, as it generates action potential, an outgoing signal, also called as nerve impulse. You probably heard this name. Nerve impulse. And this nerve impulse is conducted through this axon, and it will pass through the branching of the axon ultimately to the next neuron. So this is how the neurons are linked with each other. This is how they will work with each other. Okay. Now if we look at the direction of the flow of this nerve impulse, you will see this is another feature for the nerve impulse and the whole nervous system, is that it is directional. There should be a specific direction of the flow. The incoming signal are integrated in the some region of the dendritic cell as well as the inside the cell body. Okay, and then the action potential is conducted along the axon, which is this regions. The action potential will be turned on, and if you look at these arrows, it will give you an idea about the directionality of the flow of the nerve impulse. It generates from some point. It's received actually from the earlier neuron, and then it gets transferred through the dendrite ultimately to the cell body towards the axon, and axon will carry it to the next neuron. So this will be the directionality. It will never flow opposite. So the direction goes like dendrites, then cell body, then axon. Dendrites collect the electrical signal, cell body integrates incoming signal and pass it towards axon, and axon passes the electrical signal to dendrites to the another and the next cell, and ultimately it will reach to the effector cells. Now the signals at the end reach the end point of the axon. We call it axon terminal, where we see axons are also branched at the end. These are the axon terminal. And if you look at this axon terminal also interact with the nearby or the next neuron dendrite. Okay, so these are the regions where the axon terminal interacting with the next receptor site is known as a synapse or synapse. Okay, so here the dendrites and the cell body provide a large surface area for the communication with the other neurons. So signal from one neuron pass through this axon and ultimately reaches to the next dendrite of the next neuron. And this is the region known as the synapse, where all the process of chemical transmission continues. Okay, and this chemical transmission is very important for responsible activities in our body, because nerve controls so many different activities. It controls the motor activities, that is a movement of muscles in our body, and all these things require to release of some chemical factors, in some cases hormones, in some cases some other neurotransmitters, example acetylcholine or gamma aminobutyric acid, glycine, which act as the neurotransmitters, that is a chemical signal nerve cell can understand, and they will respond accordingly. Now another thing I should tell you about the neurons is that the length of the neuron, those things vary. Axons, especially the axons, if you look at here, the length of axons, it varies from time to time. Because the cell body cannot be so much bigger. It's kind of adjusted always. But the length of the axon, it can be huge. In some case, it can be short. Because neurons are longer cells. This is the tallest cells of our body, the largest cells. Not actually large, but we can say it's a longest extension of the cells of our body. Right? Because, they can be short. And especially when we talk the neuron is short, we are calling like the axon is short. Short axon means is just approximately, let's say 1 to 2 millimeter can be the length of an axon. It communicates only with the cells of their immediate vicinity. If a neuron has a cell body of 50 micron in diameter, for example, if you imagine 50 micron in diameter, its action would be only about 1 millimeter long. Okay. While axons can be also very long. For example, it can also be greater than 1 meter. So think about it. And communicate over the long distances. For example, the axon of some spinal cord neurons that can reach all the way to the muscles of the big toe. Just think about it. It's covering whole body of a person. It's a hugely long neuron. In general, the longest axons are associated with the large cell bodies, but still, cell body is not elongated, it's not extended. Normally, there is a relation between the cell body diameter and the length of the axon. Okay? But it's not like the cell body also will be elongated something. Now, if you look at the structure of axon, another few things you will see there. Let's look at here again. Another few things, that the signals that are sent out along the axon, like each neuron has a single axon which arise from a cell body. Because you see all the picture, it draws only one axon. They can have multiple dendrites, but axon will be only one. Now axon can branch from axon collaterals. That's what we see. At the end of each of these axon, it forms. Not only at the end, but also it can branch. Call it collateral axons. Okay. At the terminal ends, axons can branch, which we see the terminal budding or branching, which we call as axon terminal. Now, the job of axons are mostly to transmit that nerve impulse. That means to transmit that chemical signaling throughout this region, throughout the distance. Actually, it's working like a wire. If you think that that impulse is as an electricity or electric current thing, then these axons are nothing but wires. And actually they are behaving like wires, because if you look at the wire has a metallic, normally the core part of the wire is made with a metal, either copper, mostly copper in that case for a good wire. Now outside that, we insulate that thing with a coating, PVC coating or something. We insulate that thing so that the transmit of the electric current works faster and better way, and also for the safety. The same thing happens for an axon. If you look at the axon, you'll see there are a few regions in the axon which are very thinner, but there are a few regions with something like a coating going on. Okay, it's not a continuous coating, but it's a discontinuous coating. This coating is known as myelin sheath. And this myelin sheath is formed with a specific type of cells. They coat the axon. That ultimately give us small nodes present in the middle. You see the small nodes, only gaps in the middle through which the impulse can transmit. So they don't have to transmit the impulse throughout the naked axon. It's kind of covered in different regions known as myelin sheath, and there is a node formed. This node which are formed are known as node of Ranvier. Okay, so it's just like there is a wire which is covered, but few part of the wire is opened up. This is kind of the structure for the axon. Okay, now some of this axon, now this may not be true for all the axons, but there are two types of axons you will see. One type of axon will see the naked axon, which normally don't have any type of this coating or myelin sheath. So we call them non-myelinated axon and neurons. But there are some with this myelin coating. We call them myelinated axon or myelinated neurons. In those myelinated neurons, this myelin coating helps them to transmit the signal faster compared to the naked axon. So if the axon without any myelin sheath will transmit the signal, but it will take more time. If the axon contains a myelin sheath, it can transmit the signal very, very fast. As for if see here, this is kind of an actual complicated structure of the neuron. This is the picture that I want you to look. You see the dendrites are further fold, divide and branch, just like a tree and the root of the tree. Similarly, axons can also be branched, but they are known as collateral axons that I told you earlier. You see here, this is one example of a neuron. You see this is the cell body, and this is where all this coating, and if you make a cross section of the coating, you will see the transfer of this nerve impulse. It's nothing but action potential. And this potential is generated and maintained due to the opening and closing of ion channels that are present in the membrane of this axon. Okay, so all these ion channels should be present throughout this axon if the axon is non-myelinated. But if it's myelinated, then they don't need to put all this action potential movement in throughout the length of the axon. They only need to put it to the node of Ranvier. That is the reason if they have node of Ranvier, they need less amount of those membrane ion channels which can open and close based on different time. That will help to propagate the nerve impulse. That's the reason if axon is myelinated, it can transmit the signal faster, because simply the signal jumps, so it's simply jumping from one node to the other node. It's transmitting through the nodes only, so very less time to go. Otherwise it needs to propagate throughout the length of the neuron, which will be much time consuming. Now what we're talking about, these nodes or myelinated neurons, they also have a specific type of cells covering them. These are known as Schwann cells. You see, it's known as SCHWANN, Schwann cells. Now because the Schwann cells are small compared to the length of the axon, it takes many of them to insulate a single axon. That's why it forms that node of Ranvier, which is the essential element to cause the transmit of the action potential. And if you look at this picture, it will give you an idea about how this Schwann cell is insulating. It's just like the wire insulation. You see, this is the central part of the axon, the naked axon. And this is the Schwann cell that we are looking. This is the Schwann cell. So Schwann cell is wrapping around that axon. And you see the nucleus of the Schwann cell as well. Now this wrapping can be a further fold and tight wrapping. We call it myelination. The better myelinated an axon is, the better and the faster that axon can transmit a nerve impulse. You see here, you can see further structures of the Schwann cells and how they are interacting. They are just for the insulation part, and this cell is kind of wrapping this axon multiple times to form, and the nucleus is present at the top of all this myelination. Okay, and formation of these nodes, which gives them enormous capability and faster way of transmitting. Because you think our body works very, very fast with some responses, especially if you take a response against heat. Let's say you are walking barefoot and you accidentally put your feet in some hot region. Your leg will promptly lift it up. That is a very fast response for a normal person, and that amount of fast response is caused by these neurons, and the possibility due to this coating of the Schwann cells and insulation and the myelination of the neurons. So for summary, what we have understood till now, that the neurons have receptive and integrative regions, the dendrites and the cell body, which receive and integrate the incoming signals. While the neurons also have a conductive region, the axon, which generates and transmits an outgoing signal. Axons vary in length from 1 or 2 millimeter to even more than 1 meter, depending upon the purpose and the region of the distance it needs to cover through the body. Some axons are insulated with myelin sheath. Now, when they are insulated, they can transmit the signal faster. So thanks for watching this video. Stay tuned to the next video so that you can understand about the process of the transport of the signal as well as action potential and ion channels. If you like this video, please hit the like button, share this video with your friends, and definitely subscribe to my channel to get more videos like that. Thank you. We've been talking about the nervous system, and this is a series of videos when we talk about the different stages of human nervous system. And in human nervous system, the last video we talked about the nerve anatomy, and this video will be talking about the ion channels. Ion channels play a very important role in generating and propagating action potential, which we also call as a nerve impulse. Okay, so we'll understand the nerve ion channels first, then in the next videos we'll talk about how this action potential generated and how it's propagated. Okay, so let's talk about it. What are ion channels? Ion channels control the movement of ions across the neural membrane. These tiny anatomical structures make neuron excitable because the typical functionality of neurons that we know of is transmitting signal and transmitting nerve impulses from one region to the other of our body, and that thing is only possible due to the constant opening and closing of these ion channels that are embedded in the membrane of axon. So our goals for the learning for this section is to understand what ion channels are, to learn where the ion channels are located, and to understand how ion channels function and the different types of ion channels based on their functionality. Let's look at the ion channels that are present. Ion channels are normally located in the cell membranes of the nerve. So let me take a color first. They are located in the cell membranes of the nerve or neurons. So neurons mostly have these ion channels in their axon. If you know the anatomy of a neuron, which we talked in the last video, you know that axons are the extension of cytosolic domain, which helps to transfer the nerve impulse to the nearby neuron, or it can release the neurotransmitters in the neuromuscular junction. Now, if you recall the cell membrane structure, it's a lipid bilayer in which the large protein molecules are embedded in the membrane. Okay, there are integral proteins. And there's a transmembrane proteins. You know the transmembrane protein, if this is the cell membrane, and the job of a transmembrane protein is to properly embed itself in that membrane. So it totally embedded to the membrane from both intracellular side to the extracellular region. So it's connecting both the regions together. And those cells, those proteins, they sometimes can make channels. Channels are small openings through which molecules can go in and out. So ion channels control the movement of ion, the net flow of ions, that can be positively charged ion or negatively charged ion, across the membrane. These channels can be of different type. These channels can be selective. That means ion channel can control which type of ion will migrate and move. For example, let's say an ion channel that only allows sodium to pass, not any other. It will not allow potassium to pass to the same direction, because it's not about the selectivity but also the directionality that plays important role, whether the ion is going inside the cell or going outside the cell. It can also have passive or active, either of these two types. For example, in some cases of ion channels are normally present and the molecules can pass down their concentration gradient. For example, let's say this is a sodium channel, and the sodium concentration is high outside the cell and low inside the cell. In that case, sodium will go down the gradient from high to low through that channel, and that will not require any energy. In some case also, ion channels require energy from ATP hydrolysis for the transfer of ions across the membrane. For example, sodium potassium ATPase pump that act as an active ion transporter. This can be regionally located. That means ion channels location can be of very important role in case of the nerve impulse and transfer of the neural informations. It can be located in specific regions of the cell based on the type of effect the cell needs to achieve. And this channel can be functionally unique. Now, according to the functional uniqueness, we can divide them in many types. Some of them can be of controlled channel, for example, ligand gated channel, or voltage gated channel, or stress activated channel. And the other type of channels can be of without the gate. It's an open channel, so there is no gate to the channel. For example, we see the sodium transport. No gate is there. It's open. Molecules can pass. Aquaporin is another type, which is not an ion channel though, but it passes water across the membrane. Now, if we look at here, these all channels listed here are gated in this picture. Some of them are voltage gated. Some of them are ligand gated, some of them are stress activated. So these are unique. This is a type of unique channel when the organism is undergoing any sort of stress, the cell is witnessing any kind of stress, that will impact the channel to open up, and then only molecules can pass. On the other hand, the ligand gated channel will remain open once a specific chemical molecule, either from extracellular source or from intracellular source, will bind to the specific receptor region of the channel. That will make the channel to open up. While on the other hand, some voltage gated channel also works. Due to the change of the membrane potential, those channels opens up due to the change in the voltage in and out of the cell. So these are the different features of ion channels that we are going to see. So let's look at the selectivity of the ion channel. The channel selectivity depends on many factors. Now, the feature of the selectivity is important because this allows some ions to pass through and prevent the passage of other ions. If we look at this picture here, you will see two different ion channels are listed, and you see different ions are also listed: sodium, potassium, chloride, and A-. This is another big type of channel. For example, this ion channel is selective to only sodium, so it will only allow sodium to pass. Or let's say it's selected to potassium, so it will only allow potassium to pass, but it will not allow A to pass, because in this case A is larger one. So channel selectivity depends on three different factors. First is the charge of the ion. That is whether it's a positive or negative charge. Second thing is the size of the ion. If the ion is much bigger in size compared to the diameter of the channel itself, it will not allow that ion to pass. But if it's smaller, then it can easily pass through the channel. And third one is how much water the ion attracts and holds around it. This also provides an important role. The types of ion channel based on our understanding can be of two different types based on whether they are always open or they are closed. Now, based on that, we call it active channels and passive channels. Active channels are gated. They open only in some control. Okay, so the gate can only open or close based on specific regulation and control. If we look at here, this is one example of the active channel which is remain closed normally. Once the specific molecules bind, then only there's a signaling that allows the channel to open. If you look at this picture, it will also show you the idea. This is an example of voltage gated ion channel. It can be chemical gated ion channel. So you see, this gated channel normally contains a closure at the end that will not allow the molecules to pass. But once there is a change in membrane voltage, let's say it's a resting potential of minus 70 millivolt, it changes, it alters to minus 90, for example, that channel opens up. And then again it will come back to 70, it will close down. So normally in this situation it's closed down. So let's say it come back to 90, and that channel, you see, opens up. Okay, so it's an example of voltage gated channel. While the passive channels, also called as the leakage channels, because the leakage channel means they are always open. There is no matter whether there is a voltage change or chemical signaling, they are always open, and all the molecules, all the ions feel free to migrate across this channel, such as this one. Okay. Now, what are the different types of channels that we're talking about? The channels that we're talking about are of different type. Voltage gated ion channels. In the voltage gated ion channels, these are active channels. They have gates that can only open and close due to specific voltage. If you recall that the slightly more positive ions are present on the outside of their membrane, and slightly more negative ions are normally present inside the cell membrane. This difference produces a voltage. We call it as a membrane potential. If I draw it, this is a cell, slightly more positive charge present outside, slightly more negative inside. That is a normal resting membrane potential, that is approximately minus 70 millivolt. Now what happens? Now due to the opening or due to the change of this voltage by any means, okay, so for example, say the difference, it produces a voltage called the membrane potential across the cell membrane. When a neuron is at rest, these voltage gated channels are closed. So nothing's going in and out. Okay. Now during an action potential, this voltage across the membrane gets altered. It's modified from the resting membrane potential. And that will cause the voltage gated channels to open. For example, there is voltage gated channel for sodium, there is a voltage gated channel for potassium. And there are also normal non-voltage gated channel also for sodium and potassium. But here, when the sodium voltage gated channel opens, membrane's potential goes from minus 70 millivolt, which is the resting potential, and to get less negative value, let's say minus 90 millivolt. And this is an opposite ion, that's why moving inward, making the inside of the membrane more positive so that to keep these things in balance. Okay. Similarly, so this is an example of the voltage gated channel. You see, normally it's closed, but whenever the voltage is changing, it can be open. Similarly, if you look at the potassium voltage gated channel, when it's open, the membrane potential goes from plus 30 millivolt to more negative values, that is minus 70. This is because the positive ions is moving outward, making the inside of the membrane more negative. So the action of this sodium and potassium gated channels are working opposite to each other. While opening of the sodium gated voltage channel brings sodium inside the cell to make the inside more positive charged, but opening of potassium channel will cause potassium to flow out and restore the membrane potential and make inside of the cell more negative. The second type are chemically gated channels. The chemically gated channels are controlled by the chemical signaling molecules. Now let's look at one of these chemically gated channel. It will be more clear to understand. Now these are controlled by chemicals. Especially chemical means neurotransmitters, because as we are talking about the neurons. Now neurotransmitter example is acetylcholine, known as ACH in this picture, right? Or GABA, gamma aminobutyric acid. This is one. So there are different types of neurotransmitters that we know of. Now the idea is when the neurotransmitters bind to the chemically gated channel, as you see here, acetylcholine attaches to the specific acetylcholine binding region of the channel, they cause the channel to open and thereby only permitting ions to flow across the membrane. Okay. While if you look at here, same thing for GABA as well. GABA binding allows the GABA channel, GABA associated channel to open up, and that will allow the movement of specific ions across the membrane. Now, the functionality, however, for the interaction of acetylcholine and GABA are not the same. They function in a different way. Acetylcholine is a type of neurotransmitter. We call it stimulatory neurotransmitter, while GABA is known as inhibitory neurotransmitter, because the release of those activity of GABA will cause the process to inhibit the whole process of nerve impulse to inhibit and control the contraction of the muscle. While acetylcholine allows the contraction, GABA will cause the process of relaxation of the muscle and end the signaling process. But still, the mechanism of ion channel opening is almost the same. Now if we talk about the location of ion channels, as we till now know that they can be passive, chemically gated or voltage gated. They are regionally located in the neuron. And this regional location means is simply throughout this, throughout the axon. Now remember, if you remember the last class, we talked about that the axon can be of two different types. It can be myelinated or non-myelinated, right? So for a myelinated axon, the presence of the ion channels will be more frequent. That is, more number of ion channels should be found. While a non-myelinated, in case of myelinated one, you will see less number of ion channels. In case of non-myelinated, we see more number of ion channels. Because in myelinated, few regions are already blocked by those Schwann cells, and only roads of Ranvier are the region where the ion channels are located. Now passive channels are located in the cell membrane all over the neurons. These are the passive channels we are looking, which are not open, which are closed. Okay, so they are present all over the membrane. While chemically gated ion channels are responsible for, if you look at here, they are present on the dendrites. This is the region where they present, and they also present in the cell body, some part of in the cell body, in the cell membrane of the cell body. Okay, and voltage gated channels are the channels that are found in the axon, and especially the axon hillock, that is at the end of the axon there. So that is the position of all those channels. Okay, passive is everywhere. So passive is all over the place. Chemically gated channel, dendrite and cell body. Voltage gated in axon. This is the way. So now for summary, what we can look. we already see three types of ion channels based on their functionality. The first one is the passive channels, which are responsible for the resting membrane potential. Example of such passive channel is they don't contain any gate. It can be sodium channel, potassium channel, or chloride channel. Second one, chemically gated ion channels. Chemically gated channels are responsible for synaptic potential and the incoming signals to the neuron. That's why they present in the dendrites as well as in the cell body. They are much required for the incoming of the signal. And these passive channels are mostly required for maintenance of the membrane potential. Okay. The third type, voltage gated channels. Voltage gated channels are responsible for generation and propagation of action potential, the outgoing signals of the neuron. So they are required for induction and propagation of action potential, and that's why they present in the axon, because axon is responsible for conducting and transferring all those impulses to the nearby neurons. So these are the functions of all these three types of ion channels. Now if we look at the summary, what we can say that we've learned that integral membrane proteins containing watery pores from channels through which ions can move. Some ion channels called passive or leakage channels, which are always open. Some ion channels have gates that open and close, permitting ions to pass through them only under certain conditions. And regionally located ion channels are responsible for the resting membrane potential, synaptic potential, and the generation and propagation of action potential. We've been talking about the nervous system of human body, and we're looking at the physiological aspects of the neuron. We saw the anatomy of the neuron, and we also saw the different ion channels that are present across the neuron. I told you in the last video that the ion channels play a vital role in maintaining the membrane potential, that is a voltage across the cell membrane of a cell. Now all the cells, especially if you look at for the neurons, they have a resting membrane potential, that is a baseline of membrane potential to maintain. Now gradients cause the ions to move across the cell membrane. Like normally ions move from the high concentration to the low concentration. So due to this gradient concentration change, the movement of ions are also varied. That creates this voltage across the membrane. For example, if I draw it here, if this is the cell membrane inside and outside, and if there is a net flow of sodium ion outside the cell, there should be a positive charge outside the membrane and there should be slightly negative charge inside the membrane. So these are the type of modification that take place. The electric potential is called the membrane potential, that is formed due to the distribution of those ions, which is the result of the net movement based on the gradient of the ions across the membrane. For example, normally if you look at these nerve cells, you'll find out that there is a slightly positive charge outside the membrane and slightly negative charge inside the membrane towards cytosol. Due to this net change, there is a electric potential formed across the membrane, and that is normally belonging to minus 70 millivolt. That is known as a resting membrane potential. Now the question is how this resting membrane potential is established. As I told you, it's due to the opening and closing of ion channels. But obviously we need to go into the details of how ion channels open and close to produce this minus 70 millivolt membrane potential. Now the question number 2 is how the cell maintains this minus 70 millivolt of membrane potential. So these are the two things that we need to learn, because we know that in nerve, when there is an alteration of this membrane potential, that allows the information to flow from one neuron to the other neuron. For that, this membrane potential is altered. is changed from minus 70 millivolt, let's say it can be changed to minus 90 millivolt. That will also allow the membrane potential to change, and that will involve in transferring the nerve impulse. So let's look at the process. So the goals for our learning of this chapter is to know the relative concentration of ions inside and outside the cell, to recognize that cells have selective permeability of ions, and to understand the equilibrium potential for potassium ion, to understand that sodium and potassium determine to control and start the resting membrane potential, and also to understand the role of sodium potassium ATPase pump that maintains the resting membrane potential. So let's look at each of this process. Start with how the membrane potential originate in the first place, and secondly, how to maintain the membrane potential. To understand membrane potential, the first thing we need to know is about the ion concentration and the selective permeability of a cell membrane. The first thing we'll understand is the change in ion concentration across the cell membrane. This change in ion concentration actually drives the movement, the net movement of ions. If you recall earlier, we talked about the intracellular concentration of sodium, potassium and chloride differs from the concentration of these ions that are present outside. Inside the cell, the concentration, if you look at here in this picture, this is inside, this is outside. So my right hand side here in this picture, the intracellular side of the cell, they normally contain the concentration of the positive ions are high. It is balanced by high concentration of negatively charged proteins or other anions. Negatively charged proteins can be this bigger one, or it can be anions like chloride. It is balanced in this fashion. While in extracellular fluid, if we look at this side, the concentration of positive sodium ion is high. It is balanced by high concentration of negative chloride ions. Okay. So potassium is more inside the cell, sodium is more outside the cell, and this distribution of ions are balanced by anions that are there. So anions are not the key players that alters the membrane potential, but that helps to maintain the ion gradient across the membrane, which is also very important stuff. Now, alongside of this gradient change, we also need to understand another important fact. That is the cell membrane's feature of selective permeability. Ions are not soluble in the lipid bilayer, so they cannot easily diffuse through the membrane. They need channels or helper proteins that will allow them to pass and move. If a cell has channels for a particular ion, we say it permeable to that ion. Since most cells are permeable to some ions but not to others, they exhibit what is called as selective permeability. For example, one ion channel, that is sodium channel, it will allow sodium to pass but not potassium to pass, right? So a cell contains more sodium channels will help to move and migrate sodium across the membrane. The other cell lacking sodium channels will not be able to do so. So that is the selective permeability. Now the cell is permeable to potassium. Many cells in the body is selectively permeable only to potassium, because it's a must to have a permeability to potassium and sodium normally to maintain the resting membrane potential. Now excitable cells are very permeable to potassium. If a cell is excitable, that should be very much permeable to potassium and slightly permeable to sodium. And if you look at neurons, they are selectively permeable to potassium. So in case of neurons, if you look, they're selectively permeable to both potassium, sodium and chloride ions. As we saw earlier in the picture, you see, the gradient of all these ions are listed. While sodium, more outside, less inside. Potassium, more inside, less outside. Chloride, more outside, less inside. Cells are impermeable to any kind of negatively charged proteins or other large anions found inside them. These ions are too large to pass, so that is another way of selective permeability. This picture actually depicts the selective permeability very well, because lipid soluble molecules can easily diffuse through the lipid bilayer without any issue. They don't need any extra help or any other carrier or channel proteins to present. But non-lipid soluble molecules are prevented normally to diffuse to the lipid bilayer. But non-lipid molecule can also pass through the membrane if they have a membrane channel. That is the importance of a membrane channel protein. And we've already talked about the membrane channel protein in the last video. So if you don't know about channel proteins, I should strongly recommend you to go and watch that video so that you get much idea about this video. Let's move to the fact where the permeability depends. So the permeability depends on the number of ion channels that are present, and especially the two important facts the permeability of cell is particularly depend on. First one is the number of channels for that ion. So permeability can be increased by increasing the number of ion channels that are present in a membrane. For example, if you are looking at the permeability of a potassium ion, and if the cell increase the potassium ion channel in the membrane, that cell will be more permeable to the net movement of potassium pretty easily, right? And the second important factor is the ease with which the ions can move through the channels. If an ion is small compared to the size of an ion channel, it goes through easily. But if it's a larger one, it will be trapped. And not only about the ease, because there are two types of ion channels. One is passive, which we are looking in this side, don't have any gate. Another one is the active. Ion channels contains gate that needs to be opened so that the ions can move. All these factors control the movement of ions across the membrane. Now this permeability can be changed rapidly when they are gated or by any means of gating. For example, they can be voltage gated, or they can be ligand gated, or stress response channels. The permeability of a cell for a given ion increases when gated channels for that ions open. This is the mechanism used for the nervous system to produce rapid change in the membrane permeability. Because in neurons, to transfer that signal from one neuron to the next, they need to alter the resting membrane potential, and it generates what is known as action membrane potential. The action potential is generated so fast, and the only way possible is to open all those gated channels, especially the especially the voltage gated channels, which are present throughout the axon and axon hillock. Now let's look at the process of the resting membrane potential induction, how it starts. The first thing we talked here is about the change in the membrane permeability and how the membrane permeability will be changed. Well, the first important stage for the generation of membrane potential is the event known as potassium diffusion. So let me change the color once again, otherwise it should take a dark color. This is black, yes. So the first event we can say is the potassium diffusion. Potassium diffuses down its concentration gradient from the cell outside. We know that cell contains more potassium inside and less sodium inside. So if we look at here, we are studying neurons, but before we talk about how ions move across the neural cell membranes, let's talk about the simpler cell, one that is permeable to only one ion. Many of the cells of body are like this simple cell, non-excitable cell. When we understand that, we will learn how the neurons and they function, because they have a different speciality of that function. Now let's say this cell is selectively permeable to only potassium. We are talking about this cell. So selectively permeable to only potassium. So only potassium can move out and in, nothing else. Okay. Now gradients cause ions to move. Potassium will diffuse down its concentration gradient from the area where the concentration of potassium is high to the area where the potassium concentration is low. Now normally potassium concentration is high inside the cell and low outside the cell. So that's the reason potassium will move from the cell outside. So it will diffuse out. Okay. So as this concentration gradient acts as a chemical force that pushes potassium out of the cell, if you look at here, this indicates the direction of the chemical force which is non-excitable cell receives in. So normally a simple movement of it. So if this is the cell we are looking at, and if we think that this blue colored ion is the potassium, we can say that this will move, diffuse out of the cell. This is the event. Now after this event there are some chain events associated. Diffusion of this potassium out of the cell creates an electrical potential across the cell membrane. Do you know why? Because as potassiums are positively charged, movement of all these potassium ions outside the cell cause a net positive charge outside the membrane and a net negative charge inside the cell or cytosolic region. So this separation of charge creates an electrical potential across the cell membrane, which is also provided by this picture. You see mostly negative side in very close proximity of the cell cytosol and very positive charged surrounding the cell outside. Now this electrical potential will start opposing the diffusion of potassium out of the cell, because once this process starts to occur, then what will happen? Sooner potassium concentration will be higher outside the cell and it should be lower inside the cell, right? So if you think of this idea, that imagine that we put our cell in a bath of extracellular fluid and watch potassium diffuse out. As potassium diffuses out, an electrical potential begins to develop. Now since opposite charges attract each other, the developing electrical potential begins to pull potassium back inside the cell. Because you know, potassium gradient is very high now outside and very low inside. So again, based on the same rule, potassium will tend to flow inside the cell and this process will continue to occur, continue to go, right? This width of the arrow, if you look at here both the arrow, the width of the arrow, it represents the strength of electrical forces that we are looking. The potassium continues to diffuse down its concentration gradient. The developing electrical potential or force start to increase. Okay. Potassium will continue to diffuse out until the electrical potential is equal but opposite to the force from the concentration gradient. Two forces act on potassium together. These forces are called as the electrochemical gradient. So let's look at that process. These two forces that we're talking about that controls the movement of potassium. One is the concentration gradient or chemical force that causes potassium to diffuse out of the cell. And the second one is the electrical potential or electrical force. This electrical potential originates after the net movement of potassium out and the net change of the membrane potential. And that pulls the potassium inside the cell. And since these chemical and electrical forces on potassium are equal and opposite, there will be no net movement of potassium across the cell membrane. So we can say that potassium here is at equilibrium. So no net movement. The amount of potassium moves outside, the same amount is coming back inside. The force allow them to move outside is the concentration gradient, and the force that will bring it back is the electrical potential or electrical force. Now if you look at the equilibrium potential of the potassium, what we will see? That this electrical potential across the cell membrane is called the membrane potential. That is what known as the membrane potential. That keeps the membrane in a specific shape. The membrane potential is measured in millivolts. Okay, it's a simple voltage, so we'll measure it in millivolt. Because the inside of the cell is negative, the membrane potential is also negative. value. And this negative value responds for different cells differently. For example, as we've seen, a simple non-excitable cell has a concentration of 150 millimolar potassium inside the cell, and this is outside the cell, okay, and 5 millimolar potassium only outside the cell. At these concentrations, the chemical and electrical forces on potassiums are equal and opposite when the membrane potential is balanced at 90 millivolt, minus 90 millivolts, because it's ultimately negative value. Okay. At this spatial membrane potential, the potassium is at equilibrium. This is known as the equilibrium, and it's known as the equilibrium potential for potassium. Similarly, we only saw this example for a non-excitable cell and for only potassium. The same thing can happen also for sodium. The same thing we can also look for chloride, any other ions. Okay, but for the non-excitable cell. But a cell that is excitable, in that case, the situations will change. And then we'll see what is the main thing. Now we'll see the electrochemical gradient for sodium and how this thing works. Okay, so let's look at it. We'll recall that the neurons are permeable to more than one ion. So instead of only potassium, we also know that it is also sodium is also permeable. So let's see how the membrane potential in such cells differs from the membrane potential in simple cells that are only permeable to potassium. So when neurons are not generating electrical signals, we said that they are at its rest. They are not generating any signal, but still they are maintaining their membrane potential, right? So resting neurons are very permeable to potassium. So if you look at here, resting neurons, the permeability, let's say this is the permeability high and low. They are very permeable to potassium and only slightly permeable to sodium. They are also permeable to chloride, but since it contributes a little to the resting membrane potential, we should not consider chloride in this purpose. We only consider to talk about potassium and sodium. So we have observed that the movement of potassium ions across the membrane, it's a very permeable cell membrane. Now let's examine the process of movement of sodium. So now if we look at this picture, you will see the arrows are also similarly distributed, and the width of the arrow provides the degree of this potential in the movement. Okay, the relative strength of the chemical force. So till now we have learned that the potassium diffuse out of a cell, a charge separation develops. The charge separation is positive outside, negative inside. That is due to the potassium. We saw that. Now, since opposite charges attract each other, the chemical potential resulting from the charge separation, and also we know that the potassium seriously flows in back there. Okay. Now here you see that the width of the arrow again representing the degree or the strength, the relative strength of the electrical force that is there. Now the force from the concentration gradient and the electrical potential, they combines to produce a large electrochemical gradient for sodium. So what happens for the sodium? Okay, let's look at it. Normally, the potassium diffuses out of a cell, charge separation develops, producing a net negative charge inside the cell, net positive charge outside the cell. Neurons are very permeable to potassium, so they have a net negative charge inside their membrane. Since opposite charges attract each other, here the electrical potential resulting from the charge separation act as a force to pull sodium also, not only potassium but also sodium inside. That is the simple difference that we see in case of neurons. If a cell is only permeable to potassium, then only potassium should flow, But now we know that not only potassium, but here a lot of sodium is also present outside, and we know that sodium concentration is more outside. So sodium will also flow in along with potassium inside the cell. Okay, and here you see the width of the arrow representing the relative strength of the electrochemical gradient. So we see the electrochemical gradient is very high for both sodium and potassium. That will rush the sodium and potassium inside the cell. This electrical gradient drives the sodium. Okay. Now if you think of the resting membrane potential caused by both potassium and sodium movement, then what we will find out? In neurons at the rest, the membrane potential is called as the resting membrane potential. So if a neuron were permeable only to potassium, its resting membrane potential would be normally minus 90 millivolt, just like any other normal cells. However, the resting potential here also slightly permeable to sodium, and the electrochemical gradient that we saw for sodium, it also very high, that will move the sodium also inside the cell. The resting membrane potential results from the movement of both sodium and potassium ions, and both of them are positively charged ions, right? So the potential here will be little bit positive than minus 90 millivolt, because now not only potassium is moving in, but also sodium is moving in, right? So it shifts this resting membrane potential from minus 90 to minus 70 millivolt. Okay. So if we look at here in experiments, if you look, normally in case of cells which is only permeable to potassium, it should be minus 90 millivolt. But if it's a nerve cell, that case, while the electrochemical gradient is pushing potassium, not only it will put potassium, it will also push sodium. That will change the resting membrane potential from minus 90 millivolt to minus 70 millivolt. Okay. That is how this resting membrane potential will be originated. Now the question is maintenance of this resting membrane potential. You see here, it's noted that this potassium, it's relatively large net diffusion of potassium out. And then while we are taking it in, not only potassium, but sodium is also taking inside of the cell. So we call it a slight modification. Okay. So what we say in case of nerve cell, the potassium leaks out of the neuron and sodium leaks in. Okay. This is how they maintain the resting membrane potential always. When the resting membrane potential is not equal to the potassium equilibrium potential, the forces acting on potassium are no longer equal to the opposite. Because you see at minus 70 millivolts, if we see at minus 70 millivolt, the chemical force pushing potassium out of the cell, so potassium out of the cell is greater, much greater than the electrical force pulling potassium in. Electric potential, and this is the gradient, right? Now force on potassium is small, but the neurons is very permeable to potassium. As a result, still small amount of potassium moves continuously out of the neuron. But at 70 millivolt, the same situation, but the force on sodium is very large, right? The force on sodium is very large, but the neuron is only slightly permeable to sodium. As a result, a small amount of sodium moves continuously into the neuron. Small amount, but it will move continuously inside the neuron. Okay. So at minus 70 millivolts, the resting membrane potential, if you look at the resting membrane potential at minus 70 and the scenario of both potassium and sodium and what they are doing, you will find out that potassium leaks out to the neuron a little bit, and similarly sodium leaks inside the neuron a little bit. The reason behind it is the permeability of potassium and sodium. Okay, because potassium is forcing inside because of the electrical gradient, the large electrical gradient, and sodium is going inside due to the large electrical gradient, and the potassium is moving outside due to the higher permeability. Okay. Now, if you look at the same thing listed here, so this is how they keep maintaining the membrane potential. Whatever is generated, that is minus 70 millivolt, it's generated. And once a cell reaches this minus 70 millivolt, we know due to this change in the gradients as well as the electric potential, there is always a net slow movement of potassium out of the cell and slow movement of sodium inside of the cell. Now, this is how they balance the membrane potential. But while they need to generate any action potential, they utilizes different types of gated channels. For example, voltage gated channels. You see, whenever they change the voltage gated channels and modify it, it opens up for specific ion only, and that will rapidly change the membrane potential, and that's how the action potential actually originates. We will talk about the action potential in much details in the next video, but for now you should know that the last part of this process is the neuron must compensate for the potassium and sodium leak, and how they compensate for the potassium and sodium link, and that thing is compensated due to the redistribution of sodium and potassium across the membrane. Because if you look, normally just as a boat that begins to leak will eventually sink, so a leaking neuron will eventually fail to its function. So if the ions leak continues, in that case the concentration gradients of sodium and potassium will decrease. As the concentration gradient decreases, the membrane potential moves towards zero. So when there are no longer any chemical or electrical forces to move ions across the membrane, the neuron cannot send or receive any electrical signal. It needs to communicate. So the captain can keep her boat afloat by simply say belting water out fast as it can, and as fast the water enters. So neurons can prevent the potassium and sodium gradients from running down by transporting potassium back into the cell and sodium back out of the cell. So remember these things. So to prevent this leak or leakage of ion, so the cell needs to transport potassium inside the cell and sodium outside the cell. But the question is, we know that this is against the concentration gradient. Normally, the concentration gradient favors the opposite directionality. So how can cell manage to do that? The answer for that is that cell utilizes a specific ion transporter known as sodium-potassium pump. This sodium-potassium is also known as sodium-potassium ATPase pump. This is the pump which utilizes energy from ATP hydrolysis, help to maintain this minus 70 millivolt of nerve membrane potential for neurons, and also that helps in maintaining the net integrity and the concentration of ions across the membrane, so that the whole process of membrane potential continues. Now, the job of this sodium potassium pump is simply to normally, you see, this is a pump that can bind with both sodium as well as potassium ion. It can also interact with ATP. So two molecules of sodium binds to the specific sodium binding site, and then it will also interact with ATP, and then ATP hydrolysis cause the transporter to open up. Actually, I think 3 sodiums are attached, not 2. So it will open up, then the sodiums are transported out, then 2 potassium will interact, and the release of the phosphate group from the pump cause another structural shift that allows the potassium to flow in. So first the ATP attachment caused the structural shift that caused the sodium to move out. Then the second one, once potassium attached, you see these are, yellow is the potassium and sodium is denoted with this red one. So once potassium is attached, they undergo another structural shift. The phosphate group is released, and that will cause the movement of potassium inside. So this sodium potassium ATPase pump balances the concentration of sodium and potassium across the membrane and reshift the concentration, says potassium ion concentration is higher inside the cell and sodium is higher outside the cell. That's how they like it to maintain. So that in a sense how the membrane potential is generated and maintained. Now if we look at the summary, the concentration of sodium and chloride ions are high outside the cell in the extracellular fluid, and the concentration of potassium and organic anions are high inside the cell. The permeability of a cell for ions depending on the number of type of ion channels in the cell membrane. The electrical and chemical forces for a particular ion combines to become a single force, that electrochemical gradient, which cause the movement of ions across the cell membrane. In simple ion excitable cells, non-excitable cells for example, the membrane potential depends only on potassium. Potassium comes to equilibrium when the membrane potential for the cell becomes minus 90 millivolts. The resulting membrane potential in neurons depend on the distribution of sodium as well as potassium across the cell membrane, and the resting membrane potential in the neurons are commonly around minus 70 millivolt. The sodium-potassium pump is essential for maintaining the resting membrane potential in the neurons so that it balances as the resting membrane potential keeps minus 70 millivolt range. We've been talking about the human physiology lectures, and we've been talking about the neuron and the nervous system of human body. We've talked about the neuron anatomy, and we also talked about the resting membrane potential and how the cell maintains resting membrane potential. In this video, I'll tell you exactly how the nerve cells communicate with each other through the process of generating nerve impulse. So the question is, what is that nerve impulse? Remember, we already heard this name a couple of times. So what is nerve impulse? And how this nerve impulse is generated and how it's propagated? Nerve impulse is nothing but the change in the membrane voltage. Normally, all the cells, they have a change in the current inside and outside of a cell membrane. Okay. Usually it's slightly negative inside the cell and slightly positive outside the cell. We call it as a resting membrane potential. But this resting membrane potential gets altered due to the opening and closure of certain ion channels that are present in the cell membrane. Due to that modification, this resting membrane potential is also altered, and that we call as an action potential. Neurons communicate over long distances by generating and sending these electrical signals. These are known as action potential or nerve impulse. So the goals for our learning is to understand that the rapid changes in permeability of the neuron membrane produce the action potential, to recognize that altering voltage-gated ion channels change membrane permeability, to understand that the movement of sodium and potassium ions during the action potential, and to examine the refractory periods. And what do we mean by refractory period? And to also learn about the conduction velocity and what are the factors this conduction velocity or the nerve impulse ability rely on. So let's talk about the overview of the action potential. The action potential is a large change in the membrane potential from a resting value, which is in case of neuron is minus 70 millivolt, to peak about plus 30 millivolts, and then again it will come back to minus 70 millivolt again. The action potential results from a rapid change in the permeability of the neuron's membrane for two different ions. One is sodium, another one is potassium. The permeability changes as the voltage-gated ion channels open and close. Remember, if you saw my earlier video regarding ion channels, I talked there about the different types of ion channels. There are voltage-gated, ligand-gated, and also there are passive ion channels. Passive channels are always open. But these voltage-gated or ligand-gated or stress-activated ion channels are open only for certain amount of work. For example, voltage gated sodium channel and voltage gated potassium channel will open if there is a sudden change in the membrane potential. And that is the exact case what happens in case of the generation of action potential. If you think the same thing for synaptic transmission, we will see that there is the importance of chemical activated or chemical gated ion channels that plays a vital role for the synaptic transmission or the transmission of the nerve impulse between two adjacent neurons. Now, in the following slides, we are going to talk about sequential stages of this action potential. Here also, you see the different stages of the action potential. I listed the stages based on their occurrence. As this action potential is a phenomena, you know, this action potential or the nerve impulse, it starts from the junction where the cell body of the neuron is connected to the axon, known as the axon hillock. And then it continues to propagate till the axon terminal. So it actually propagates throughout the axon. Now the thing is, ion channels are embedded throughout this axon. So those channels are open and closed, so the action potential continues to move. The sequential events of the action potential follows as this. The first one is the application of adequate amount of stimulus that will change the resting membrane potential. The second event is the depolarization event. It's nothing but a sodium influx. That means the rapid movement of sodium inside this neuron. And this depolarization event goes by the same way. When there's a rapid movement of sodium, the inner charge, which is supposed to be minus, is going to be positive. Third step is known as the repolarization. When as the membrane gets depolarized, all those positively charged potassiums start moving outside, cause a potassium efflux. Then you see repolarization take place. So as the sequential ranges or different stages or as we go on from axon hillock towards the axon terminal, you will see the sequential event continues throughout the segment of the membrane. As you see it, for the first segment it caused depolarization first, then the second stage is repolarization in that specific region. But the region after that caused a depolarization. So in the next stage you will see a repolarization here. The fourth step is a hyperpolarization. Why? Because of the efflux of more and more potassium ions there. So again, there should be hyperpolarization events. Now if you look at how this signal is propagated, you will see it clearly that the events are continuously occurring in a segmented fashion throughout the axon. And after again the hyperpolarization is conducted, it is coming back to the resting state using sodium potassium pump. It balances everything out. In case of resting membrane potential also, we saw that the sodium potassium pump balances the resting membrane potential. So it again restores everything back so that the process can start from again, and those neurons can also start transmitting the signals. Now this is the graphical representation of the events that are linked with each other. And if you look at here, it starts with several different phases. Remember, the main 4 phases are out there. Resting state, where there is no change. Second is the depolarization state. Third is a repolarization state. And fourth is again another hyperpolarization state called as undershoot or hyperpolarization. Okay. And if you look at here in the y-axis, it says the membrane potential value. While we are going depolarization, potential value gets altered in the positive value. Repolarization means it's going back to negative again, and hyperpolarization is further negative. So whenever you see membrane polarization, that gives you the idea that the membrane potential is going to be negative. And whenever you heard the name depolarization, that is the membrane potential is going to up to give a positive value. So we'll see this graph throughout the sequential events and how this thing works. Look at the first process and the first step, that is the resting state. And this action potential begins at the axon hillock. I told you earlier the axon hillock is the region where the axon is interacting to the cell body. That's the junction of cell body with the axon. This action potential begins when the signal from the dendrites and cell body, they start migrating and reach this axon hillock. Until and unless they reach the axon hillock, the signaling will not start. And this movement of signal cause the membrane potential there to become more positive. This is known as the depolarization, and this is where the process will actually begin. Okay, so let's look at the processes of how depolarization occurs. If you look at this axon hillock, you'll see that there is a slight positive charge inside the cell start to generate due to the movement of sodium inside. That is the actual event of depolarization, the net movement of sodium ions inside the cell. If you look at this graph, we are going to see this graph throughout this complete PowerPoint show. In this graph, all those events are listed in the bottom and also the major events for all those things. While in the resting state, sodium and potassium channels are always like voltage or non-voltage sensitivity. They are always open in that fashion to maintain a resting membrane potential. Now, if you want to learn about how cell manage to maintain a resting membrane potential of minus 70 millivolt, I'll recommend you to watch my video on resting membrane potential. Now, the stage of depolarization, the major event is the net movement of sodium inside the cell while the sodium pump or the sodium ion channel is open. So as the axon hillocks depolarize, the voltage gated channels for sodium opens rapidly and increasing the movements of permeability of the sodium inside the cell. Normally the sodium channels are closed, but whenever the axon is depolarized, these are known as the voltage gated sodium channels, and due to the depolarization of the axon hillock, they have a slightly positively charged generated over there. That will cause this voltage gated channel to open. Once the channel is open, sodium can easily flow inside the cell. So this is outside and this is inside the cell. Okay. So if you look at here in this sequential stages, then after a few hours, after a few minutes, not actually hours, sorry, very few seconds even, when the sodium start entering inside, then again the sodium channel can be closed. So this is the stage of depolarization. Now the question is how long this process will continue, because we need to prevent this process after some time, otherwise it can create problem, right? More sodium start to flow inside, so it will cause the potential to change rapidly. So how to prevent it and how to go back to the cycle of repolarization at the end. Now the answer for that normally here is that there is a threshold. If the stimulus to the axon hillock is great enough, the neuron depolarizes by about 15 millivolts and reaches a trigger point that is known as the threshold. That threshold is the depolarization of 15 millivolt at least. So if the depolarization is not enough to change the membrane potential to 15 millivolt, then normally the process of sodium movement will not happen. At this threshold, an action potential is generated. Weak stimuli that do not reach the threshold will not produce any action potential. So action potential always have the same amplitude and the same duration. At the minus 55 millivolt that we are looking, normally it's minus 70 millivolt. I call like there's a change of 15 millivolt, so it gives us minus 55 millivolt. That is the membrane potential in the depolarized threshold that allows the action potential to start. Otherwise, action potential will never start. So even if there is a stimulus, but it's very low, that is not causing the membrane potential to be minus 55 millivolt, it will not allow the action potential to begin. Now as it begins, sodium start flowing inside. Now we have a process to stop that. But what we know is that the sodium flowing inside work as a positive feedback loop. So let's look at that feedback loop. What is going on? This is a rising phase of membrane potential from minus 55 to plus 30 or 35 millivolt, very rapid change due to the movement of sodium inside the cell. Now during this rising phase, what we know is that the depolarization triggers the sodium channel to open. That allows sodium to enter inside the cell, right? And this, when the sodium start entering, that will help more depolarization, and more sodium channels open, more sodium start entering inside. So this is the positive feedback loop caused by the depolarization. Now to prevent this, cells need to do something, right? So how cells interrupt this positive feedback loop? There are two ways to actually interrupt this positive feedback loop. One is the interaction of the voltage gated sodium channels, and second is the opening of the voltage gated potassium channel. So we either inactivate the voltage gated sodium channel, because if we inactivate this channel, if we block it, then there is no way sodium is going to flow inside. That is one thing we can do. Or another thing cells can do is open a channel which allows positively charged ion to flow outside the cell to balance the situation. Because right now, if this is inside the cell, this is outside the cell, sodium is going in, so positively charged is building up inside the cell. So if we can open up a channel that will push a positive ion outside, let's say that is potassium, that can balance the membrane potential. So that's what we're trying to do. So these voltage gated sodium channels have two gates. A voltage sensitive gate that opens as the cell is depolarized, and a second time sensitive interaction gate or inactivation gate that stops the movement of sodium through the channel after a specific time. So there is two different blocks, not only the voltage sensitive channel, but if you look at the sodium channels, if I draw it here, two different channels are there, voltage gated and time sensitive. I think in the last slide we talked about, in the last one slide we talked about that. It's not here anyways. Here you see these are the two different gates. One is a voltage sensitive gate, another one is the time sensitive gate. So the idea here is, even though the voltage sensitive gate is not working due to any means, we can simply alter, and that time sensitive gate will always work, because if voltage gate fails, time sensitive gate will work, because after certain time anyway that channel is going to be stopped, right? So at this resting membrane potential, the voltage sensitive gate is closed. As the neuron depolarize, that voltage gated or voltage sensitive gate start to open. At a fixed time after the channel opens, it inactivates. So the idea is, normally the channel will be closed, normally the sodium channel will be closed. If there is a change in voltage, it opens up, then again after a certain time it's going to be closed again. So that is the way to prevent the positive feedback loop. And the other way also, the rapid movement of potassium out of the cell, right? So here what happens after that, the second step is the repolarization. And this is the phase where we see that the potassium channels start to open up, so that potassium start flowing outside. Because we talked two different possibilities, either closing of the sodium channel or opening of the potassium. Now opening of the potassium channels will also work every single time to make the membrane repolarize and reshifting the membrane potential back. So here you see the potassiums leaving the cell as voltage gated potassium start to open. Okay. So these are the sodium channels, normally take sodiums out in, then after some times when the potassium channels start to open, sodium channel can be closed and potassium start flowing outside. It kind of restores the membrane potential for a while. Now with less sodium moving into the cell and more potassium moving out the cell, the membrane potential becomes more negative and moving towards the resting membrane potential, isn't it? Let's move to the next step. The step number 4 is known as the hyperpolarization. In many neurons, the slow voltage gated potassium channels remain open after the cell has repolarized. Because the voltage gated channel, whatever we know of, there are two types: high voltage gated and slow, that is fast voltage gated and slow voltage gated. Fast voltage gated channel remains open and closed in response to voltage very fast, while slow voltage gated channels open and close very slowly, so it takes more time to remain to close, right? So it remains open for a long time. Similarly, not in all neurons, but in many cases of the neurons, if they have the slow voltage gated potassium channel, that remains open for a long period of time. Now in that case, potassium continues to move in. So as this is open, potassium continues to move out of the cell. And due to this reason of potassium moving out of the cell, that caused the membrane potential to become more negative, even more negative than the resting membrane potential. So what happened earlier, it starts with minus 70 millivolt, then it reaches minus 55 millivolt as a threshold, cause a polarization of plus 30 millivolt. Then again, movement of potassium out cause repolarization. In that case, it will cause again balanced with some sort of membrane potential, and again movement of potassium outside the cell cause it to reach further negative content. Okay, so it caused further negative membrane potential there. So this is the hyperpolarization event that occurred. And here you see the detailed activity of all those membrane potential and how that thing is maintained over the time. Starts with the rest, then depolarization, initiation of the repolarization, repolarization, and hyperpolarization. Now in the graph, if you see the hyperpolarization, it is due to the net movement of potassium out, and you see some kind of dip in the middle there. Then again it will back to the resting membrane potential, that is again minus 70 millivolt there. So the resting potential, if you see the sodium potassium, both the channels are closed, nothing is open. Now they can open or close, those passive channels can open and close depending upon whether the cell needs to maintain its resting membrane potential or not. But actually this is not a major or big event going on there. Then the second stage of the depolarization, sodium channels open, net movement of sodium inside, potassium channels remain closed. While in the repolarization step or the initiation of repolarization, you see potassium channels start to open while sodium channels start to close down. Then during the repolarization stage, you see sodium channels start to close itself. Now this closure of the sodium channel is caused by many different phenomena that we talked earlier. And during that time, all of those potassium channels are now open, because through that channel potassium will flow outside of the cell and causing membrane potential to become negative there. And then finally hyperpolarization stage, where you see all the sodium channels are closed, all those channels in both the ways are closed, and then potassium channels, few of them are open, which are slow voltage gated channels. So if we do all these things, if you see all those phenomena in the graph, you will see this is the resting state. The second step here is the depolarization state. This is at the peak or initiation of repolarization. This is the step number 4, when the graph is falling down again. This is the repolarization stage. And then again it's falling below the membrane potential, that is minus 70 millivolt. It can go up to like minus 90 millivolt. This is hyperpolarization or fifth state. Then again it will shift back to the resting membrane potential. So it's normally a resting membrane potential, then you see a spike like a graph, like a curve, and it will go on. This is the impulse that we see, and this impulse will continue from the axon hillock through the axon till the axon terminal, and this process will go on. Okay. Now the two important understanding we want to talk about here. This is about absolute refractory period and a relative refractory period. Now what is this refractory period? If we talk about refractory period, is the situation or the time durations during the nerve impulse when even though you stimulate a neuron, it cannot initiate a second nerve impulse process. So let's say a neuron is transmitting a signal, but at a specific time frame or time point, even if you stimulate the neuron again with another stimulus, it cannot transfer the impulse. That is the refractory periods. And there are two refractory periods that we know of. The first one here is the absolute refractory period. And what is this absolute refractory period? Absolute refractory period is when the membrane cannot produce another action potential because the sodium channels are inactivated for a while. Just after the neuron has generated an action potential, it cannot generate another one. Many sodium channels are inactive and will not open no matter what voltage is applied to the membrane. Most potassium channels are open. Now this period is known as absolute refractory period. Okay, and if you look at the graph, this peak situation or the time, this time from this point here till this point is known as the absolute refractory period. Okay, the neurons cannot generate an action potential because the sodium cannot move through the inactive channels and because potassium continues to move out through the open voltage gated channels, right? Because it's destabilizing, it's balancing the actual resting membrane potential. Now the second type is a relative refractory period. This happens when the voltage gated potassium channels are open, making it harder to depolarize the threshold. This happens immediately after the absolute refractory period. The cell can generate an action potential, but only if it is depolarized to a volume more positive than the normal threshold. Because the normal threshold, the value here we are seeing, the normal threshold is much below. So this part belonging from this point to this point is the time of relative refractory period. The cell has to be depolarized to a more positive membrane potential than the normal threshold to open up the sodium channels to begin the positive feedback loop so that the process of action potential propagation can continue, right? Now this length of the absolute and relative refractory periods are important because they determine how fast neurons can generate action potential. Now these things may vary from person to person, from different age differences and also the different neural diseases, these things vary. Now action potential is propagated along the axon by the same way. That is the ultimate thing and the final thing I want to talk about. After an action potential is generated in the axon hillock, it is propagated down the axon throughout the region of the axon. So whatever membrane or channels are opening and closing in the axon hillock once, they do the same thing some part next. Then you can do the same thing after that. Then same thing after that. They transfer it in a segmented fashion. Okay. Now if a neuron is myelinated, as you see, these are the myelin seed of the neurons. In that case, the action potential can transfer only between the nodes of Ranvier. That gives the process of neural impulse transmission very, very fast. While if it's non-myelinated, in that case the impulse transmission will not be that fast because it needs to travel throughout this axon and the length of the axon, right? You see this positive charge, they flows along the axon, depolarizing adjacent areas of the membrane, which reaches the threshold and generate an action potential. Then the action potential move along the axon like a wave of depolarization traveling across this whole axon from the cell body. Okay. Now the last thing I want to talk about is what are the features that control the process of this conduction velocity. The conduction velocity is the speed with which an action potential is propagated. The conduction velocity depends on mainly two things. One is the diameter of the axon. As the axon diameter increases, the internal resistance to flow the charge decreases and the action potential travels very, very faster. So the more the diameter of the axon is, the faster the transfer of the signal is. And the second thing, how well the axon is insulated with the myelin. Recall we talked about that the myelinated axons have areas of insulation interrupted by areas of bare axons called the nodes of Ranvier. Because if you look at the same structure here, those are the seeds covering and some parts are only blank, those are the nodes of Ranvier. Now in a myelinated axon, the charge flows across the membrane only in those nodes. So it's like the impulse is jumping. The action potential is jumping and skipping areas. That will be definitely faster, right? A myelinated axon typically conducts action potential faster than an unmyelinated axon of the same diameter. So if you see, if an axon is having a more diameter as well as myelinated, that will be perfect for a very fast transfer of action potential along the axon. So now let's look at the summary of what we've understood. That the action potential is an all or none event that can travel for a long distance because it is normally regenerate electrical signals across the membrane throughout the axon. When the axon hillock is depolarized to threshold, an action potential is generated, voltage gated channels open, thereby increasing permeability of the neuron first to sodium and then to potassium. Now as the sodium moves rapidly into the neuron, it produces the rising phase of the action potential. As the inward movement of sodium slows, the outward movement of potassium increases, the membrane potential repolarizes. And immediately after the action potential, the neuron cannot generate another action potential during the absolute refractory period. During the relative refractory period, it can generate another action potential only if the stronger stimuli arrive at the axon hillock. The diameter and myelination of an axon determine its conductive velocity or conduction velocity. So that in a sense are the summary of what we've understood through this video. We've been talking about the nervous system of human body, and actually we are more interested about how neuron functions. We've talked about neuron structure, we've talked about the resting membrane potential in neuron, we also talked about how action potential is generated in neuron. And this action potential is what we call as a nerve impulse that transfer from one neuron to the next, and it propagates along the axon of a neuron. Now in this video we will be seeing how exactly one neuron is contacting the next neuron. That is the neuron-neuron interaction. And not only we'll see the neuron-neuron interaction, but also we will look at how neurons are interacting with tissues like muscle. Call it as a neuromuscular junction. Because neuron can have neuron-neuron interaction between two adjacent neurons. It can have interaction with muscle known as neuromuscular junction. It will also have interaction with different glands that will regulate the secretion from those glands. So goal for our learning of this topic is to understand the detailed mechanism of neurotransmitter release, diffusion and binding to the postsynaptic receptors. To learn that the action of neurotransmitters depend on the type of receptor on the postsynaptic cell, and to review the location and the function of these neurotransmitters. This synaptic transmission involves the release of these neurotransmitters from the presynaptic cell to the postsynaptic cell. Because here we are looking at the interaction between two neurons, neuron 1 and neuron 2. Neuron 1 secretes certain chemical factors known as neurotransmitters. Once released, it will go and bind to the postsynaptic neuron. It will activate those neurons, and those neurons will be stimulated. They will be stimulated by the activity of this neuron. And by this means, this whole process of neurotransmitter release, it ends when the neurotransmitter dissociates from the receptor and is removed from the internal junction between these two neurons, known as synaptic cleft, the region between two adjacent neurons. Now normally the interaction of neuron works like this. Neurons have a cell body dendrites are attached to it, and then it has the axon. At the end it has the branches, and a next neuron begins with the dendrites. So this junction of neuron-neuron interaction goes with axon to dendron interaction. Okay, so let's look at this structure of this neural junction and synaptic cleft, because all the process of interaction will go on in the synaptic cleft, and we'll also see how this whole process works sequentially. This is the overview of the process that we're looking at. We're looking at the events of the synaptic transmission. Okay. Now the idea of synaptic transmission, if you look at this picture, this is the pre-synaptic cell, this is the post-synaptic cell, and this region in between the two is the synaptic cleft. Okay, and you know, these dots are visualized here inside the vesicles. These are neurotransmitters, and they have different functions to play. There are two types of these neurotransmitters available. One type is known as excitatory neurotransmitter, another type is inhibitory neurotransmitter. Excitatory neurotransmitter allows the next neuron to be excited, and the inhibitory one causes the next neuron to be inhibited of its functionality. So what happens here is the action potential continues to generate from the axon hillock through the axon until it reaches the axon terminal, and this is what we call it as a axon terminal. And this is the region where all those vesicles filled with neurotransmitters fuse with the axon terminal membrane that cause the neurotransmitter release to the synaptic cleft. Once they are released, these neurotransmitters can interact and bind with the neurotransmitter receptor on the surface of the postsynaptic membrane or the cell. That will cause the ultimate effect. So the series of events that we're looking here is simply start with the action potential generation, and it reaches the axon terminal. Second is the calcium channel opens. Third, once calcium channels start to flow, that cause the release of those neurotransmitters by the vesicle fusion to the membrane. The neurotransmitters crosses the synapse, which is between the two, axon and dendron. Then neurotransmitters bind to the neuroreceptors that triggers the signal in the postsynaptic neuron. These are the sequential event of the whole process of synaptic transmission. So now let's look at each of the stages in details. We start with the process when it occurs in the presynaptic cell, and the process here is neurotransmitter release, right? So what happens there? An action potential in the axon terminal cause voltage gated calcium channels to open up. As these voltage gated calcium channels open, and why they are open? Because of the action potential. Action potential cause voltage gated calcium channels to open up. Due to this opening of calcium channels, there is a calcium inflow inside the cell, presynaptic cell. That will cause all those vesicles filled with neurotransmitter to be fused to the membrane. Now, each vesicle released a fixed amount of neurotransmitters into the synaptic cleft. Now, this neurotransmitters diffuses through this synaptic cleft, and it will reach the receptor, bind to the receptor on the surface of postsynaptic cell. Now, chemically gated ion channels, due to the binding of this receptor, opens up. Okay? Because this is not about the voltage. As these channels are chemically gated, they will remain open as long as the neurotransmitter is bound to the receptor and are not sensitive to any changes in the membrane potential. So irrespective of the membrane potential, if the chemical is attached to the receptor, it will remain open. Now the synaptic current or ion movement through the chemically gated channels may depolarize or hyperpolarize the neuron. Now due to the change, whether it can depolarize or hyperpolarize the postsynaptic cell or postsynaptic neuron, there are different events and effects that we can see. The second step occurs to the postsynaptic cell. That's known as the receptor binding. When the neurotransmitter is released, now those neurotransmitters will bind to the receptor on the surface of the postsynaptic neurons. Okay? Chemically gated ion channels will open up because these ion channels are also closed normally without these chemical neurotransmitters. But whenever the adjacent neurotransmitters are attached to this receptor, they remains open. Now opening of this receptor will cause other types of ions to flow in or out. That can be sodium, potassium or any else. For example, here you see due to the attachment of this purple colored neurotransmitter to the receptor, it caused the potassium to flow out and sodium to flow in, right? Now these chemically gated ion channels will remain open as long as the neurotransmitter is remain attached to the receptor. And these are not sensitive to any changes in the membrane potential. Even though the membrane potential is changed, still this will function based on the attachment of neurotransmitter only. Now the synaptic current or ion movement through the chemically gated channels may depolarize or repolarize or hyperpolarize these neurons. This example that we see in this picture explains this whole process of how it can depolarize or repolarize, because as the channel is open, it can transfer the positively charged ions across. That can change the membrane polarity, right, and the polarization of the membrane. Now the third part of the synaptic transmission is the termination of synaptic transmission, because all this process should have to have an end. Now synaptic transmission ends when the neurotransmitter dissociates from the receptor. This is when the neurotransmitter is attached to the receptor, they make these channels always open. But whenever the neurotransmitters are released, these channels are closed. Now most often the neurotransmitter is pumped back into the presynaptic terminal and into the nearby glial cells. Here we illustrate the neurotransmitter glutamate being pumped back into the from postsynaptic to the presynaptic neuron again. Okay? In some cases you see the neurotransmitter is broken down by enzymes and the products are pumped away. So either they can be degraded or they can be pushed back from where they are released. Okay? Now the neurotransmitter acetylcholine is an example of this process. When the process is done, the neurotransmitter is broken down. When breakdown products are transported into the presynaptic terminal, they are used to resynthesize the neurotransmitter again. So simply break them down after the work, then again we take the breakdown copies and shift it to the presynaptic neuron so that they can engulf and take all those breakdown content of the neurotransmitter and can resynthesize those neurotransmitters. The neurotransmitter which has been returned to the terminal is packaged into the vesicles for storage and subsequent release in the future. Okay? By the mechanism which this neurotransmitter is returned to the terminal is specific for each neurotransmitter and can be selectively affected by different drugs. So we can use this as a drug targeting approach for a range of drug targeting and drug development. Okay. Now final thing is the type of response from the postsynaptic cell due to the attachment of the neurotransmitter. Now we have seen that the mechanism of synaptic transmission, the sequential stages, we are talking about that. If you look at the consequent synaptic activity after the release of neurotransmitters and binding of neurotransmitters, that is the ultimate goal that neurons need to achieve. Now the action of these postsynaptic cells depends on which neurotransmitters is involved with the signal. These are the neurotransmitters that we are looking. There are multiple receptors for each neurotransmitters present on the surface of this postsynaptic neuron. Each of these receptors activates a different set of ion channels. Some of the receptors may activate sodium channel, some of them may activate potassium channel. Now depending upon which type of ion channel it opens, the response varies. There are two groups of receptors and mainly we can find it out. One is a cholinergic receptor, and another one is adrenergic receptor. Let us look at different type of receptors. We will see two types of receptors. We'll see the excitatory and inhibitory receptors. One of the excitatory receptor is this acetylcholines, and we'll look at the acetylcholine as a neurotransmitter and its associated receptors. There are multiple types of receptors for each of this neurotransmitter, and they are associated with the activity or activation of different ion channels in the cell. They can activate sometimes sodium channel, or in some case some other neurotransmitter can activate the potassium channel. Now there are two groups of these receptors associated to the acetylcholine. One of the receptor is a cholinergic receptor, another one is chemical nicotine. So these cholinergic receptors bind to the acetylcholine. Cholinergic receptors bind to the acetylcholine. On the other hand, the other group that is present there, one group also bind to the chemical nicotine, and another group is there that can bind to the chemical muscarine or muscarine. So these are the three types. This is where we see that the acetylcholine receptors bind with the nicotine. This is where it binds with muscarine, and this is one is also example of a muscarine. This cholinergic nicotinic receptor, or it's also known as NACH, cholinergic nicotinic receptor, is a very well known receptor found in the neuromuscular junction. For example, where the neuron is interacting with a muscle, and the release of neurotransmitter regulates the contraction and relaxation of the muscle. Okay. At this receptor, acetylcholine acts as directly to open the ion channels, producing a fast excitatory post-synaptic potential. Very fast activity is going on there. Acetylcholine is excitatory at nicotinic receptors, and it causes skeletal muscle to contract. So this is the reason that you can, for example, you're walking, suddenly your foot, your toe is somewhat attached to, or let's say in touch with a very hot plate or something. Suddenly your hands or toe, whatever is touching to the hot plate, is opening up, it's closing down. So due to the muscle contraction, these things happen. While the other type of the cholinergic muscarinic receptors, so we call it cholinergic muscarinic receptor, all known as MACH, cholinergic muscarinase or muscarinic receptor. This is found in central nervous system most of the time, and most of the effector organs of the parasympathetic branch of the nervous system, because here acetylcholine acts indirectly in those system. Because here is the difference between the two. We have a situation where acetylcholine can act directly, which is very fast activity, and another situation where acetylcholine can act indirectly, which is very slow activity. But the slow activity can be of two different types, excitatory or inhibitory, and those things are required in the central nervous system, in sometimes to influence or sometimes to inhibit the response. Okay. Now this acetylcholine is excitatory at this muscarinic receptors and cause neurons to fire action potential and the smooth muscle cells to contract. It will cause the smooth muscle cell to contract. Okay, but this activity is little slow. But another type of MACH receptor is also found in the central nervous system and as well as in the heart. This acetylcholine acts, here in this case the acetylcholine acts indirectly and producing a slow inhibition of the postsynaptic cell. Here it calls the inhibition. In the heart, this effect decreases the heart rate. And acetylcholine is inhibitory at this muscarinic receptor and causing neurons to hyperpolarize and the heart to slow down. The action of this acetylcholine may be excitatory or inhibitory, but the effect depends on which receptor is present on the postsynaptic cell. If it is a type of nicotinic receptor, it's always excitatory and it is very, very fast. But if this acetylcholine is binding to a muscarinic receptor, there are two chances. One chance that the muscarinic receptor binding will excite, that happens in most of the cases of smooth muscle contraction, or in some case binding of acetylcholine to this muscarinic receptor can inhibit and can cause the heart rate to slow down due to the hyperpolarization. Okay. Now let's look at norepinephrine and its receptors. This is another very important neurotransmitter, norepinephrine. There are two families of receptors for neurotransmitter norepinephrine. One is alpha receptor, another one is the beta type of receptors. Each family members is different and they are identified by its letter and number. Alpha 1, for the beta there is beta 1 and 2. Okay, these are called as adrenergic receptor. Remember we talked about nicotinic, muscarinic, and this is adrenergic receptor. So all those nicotinic and muscarinic are the part of cholinergic receptor, but this is a completely different type known as adrenergic receptor. Both alpha and beta adrenergic receptors are found in the central nervous system and more importantly on the effector organs of the sympathetic nervous system. In this case, this norepinephrine acts indirectly at alpha 1. If you look at the case of alpha 1, it works indirectly to the alpha 1 receptor and it causes slow excitation. Because we know if the process is indirect, always it's slow, right? This causes smooth muscles to contract. And alpha 1 receptors are located mostly on the blood vessels as you see in this picture, which supply the skin, mucous and abdominal viscera. This norepinephrine is excitatory at alpha 1 receptors, so it excites them, but it's a slow excitation. Now, as I also told you, there are also other types, beta 1 and beta 2. So if you look at beta 1 type now, beta 1 is another receptor. And when norepinephrine interacts indirectly to beta 1 receptor, that is mostly in the heart, to produce a slow excitation. And in this case, it will allow the heart rate to increase because this is also a slow excitation. And norepinephrine is also excitatory at this phase, but slow. The third one is when the norepinephrine binds indirectly to beta 2 receptors. And that will also produce a slow inhibition now. So alpha 1 and beta 1, both the receptors when added to or attached to norepinephrine cause a slow excitation. But whenever norepinephrine is attached to the beta 2, it causes slow inhibition. This type of interaction of norepinephrine to beta 2 receptor produces slow inhibition that causes the smooth muscle to dilate. These beta 2 receptors are mostly located on the respiratory airways, blood vessels and skeletal muscle and heart, and most of the other effector organs of the sympathetic system. Norepinephrine is inhibitory at these beta 2 receptors, and as a result the action of the norepinephrine may be excitatory or inhibitory based on which type of receptor they are binding. So one thing we can say for all this type of neurotransmitter is that there is a bunch of neurotransmitter and their associated receptors are found, and those receptors are found in different regions and different organs and tissues of our body. Now how the cell will react is denoted by the receptor where the neurotransmitter is binding. A same neurotransmitter can cause inhibitory or excitatory effect depending upon the variant of receptor they are binding. And that is true for both acetylcholine as a neurotransmitter and norepinephrine as a neurotransmitter. Now if we'll talk about the location and function of these neurotransmitters, we have learned about acetylcholine and norepinephrine, and we found out that the central nervous system, and we found them in central nervous system and also in effector organs of the nervous system. Now here we will see all the other neurotransmitters of the peripheral nervous system, because that's also very important, right? So then we'll look into the central nervous system to learn the functions of these and other neurotransmitters what are working there. So you see here the effector organs of the nervous system like skeletal muscle, smooth muscle, cardiac muscle and glands. All these things are regulated by those acetylcholine and norepinephrine. But if you look at the peripheral nervous system and how these neurotransmitters are working in the peripheral nervous system, you will see that the motor neurons of the somatic nervous system release acetylcholine. Okay, and they are cholinergic. So skeletal muscles bear NACH, which is nothing but the cholinergic nicotinic receptors. So as skeletal muscles contain these nicotinic receptors, the action of this acetylcholine on skeletal muscle is direct and fast. So if we're talking about the somatic neurons, they will work really fast and they will work excitatory by the release of acetylcholine because they only have cholinergic nicotinic receptors. The first of the two neurons in the sympathetic chain, if we look, preganglionic neuron, are cholinergic. These first two neurons in the parasympathetic chain, the preganglionic neuron is also cholinergic. While the second neuron or postganglionic neuron in both the sympathetic and parasympathetic chain, all of them have these NACH receptors. Thus the action of acetylcholine on postganglionic neuron is direct, fast and excitatory. So you look at here, for postganglionic neurons it's always excitatory, it's always fast for both sympathetic and parasympathetic neurons. But for somatic it's different. In somatic we also talked it carries NACH, but postganglionic neurons carry NACH, so they will also have a fast response. We know that these somatic cells or somatic neurons connected to the effector organs like skeletal muscle, while this sympathetic and parasympathetic part of the autonomous nervous system, if you see, they are linked with smooth muscle, cardiac muscle and gland. Let's move to the next one. If we look at the sympathetic postganglionic neurons, they release norepinephrine. If we go back here, sympathetic postganglionic neuron, they release norepinephrine and they are adrenergic. Parasympathetic postganglionic neuron release acetylcholine, so they are cholinergic. Both this acetylcholine and norepinephrine act indirectly on the effector organs of the autonomous nervous system. Their action is slow, sometimes excitatory, sometimes inhibitory, but it completely depends on which receptor is found on the surface of the effector organs like skeletal muscle, smooth muscle, cardiac muscle or gland. So they can either inhibit or excite depending upon which receptor is provided on the surface. and especially which receptor is provided in the postsynaptic neurons. If we talk about the excitatory and inhibitory neurotransmitter in CNS, in this case, in central nervous system, glutamate is the most common and most potent excitatory neurotransmitter. Glutamate. Simply write it as GLU. Glutamate acts directly on the ion channels that permits the passage of both sodium and potassium movement that produce a fast excitatory postsynaptic potential. And the major inhibitory, so this is the major excitatory. So the major inhibitory neurotransmitter in the central nervous system is GABA, gamma aminobutyric acid, and glycine. GABA and glycine binding to the receptor which directly opens chloride channel, which completely reverses the mechanism of glutamate action, because chloride channel will transfer negatively charged ions across the membrane. It causes fast inhibitory postsynaptic potential. In central nervous system, we need these fast actions, because in controlling all our smooth muscles, it's always not fast. We don't require those things to work faster always. Simple modification, sometimes you also need slow modification. But in CNS, we need fast actions. That's why we have glutamate as a positively influencing neurotransmitter, and GABA and glycine as a negatively influencing or inhibitory neurotransmitter. If we look at the fast versus the slow synaptic action, there is a difference between the direct and indirect activation of the neurotransmitters. The rapid signaling of the directly acting neurotransmitter is important for the secondary motor coordination and communication and many other higher functionalities. So they help in the sensory motor coordination, communication, higher functions. For example, rapid synaptic signaling is essential for coordinating sensory output and input together. Sensory input along with the motor output, especially during the athletic performances, that helps you to do this job. Rapid synaptic signaling is essential for speech and other form of communication, and also important for playing music and requires rapid synaptic signaling. On the other hand, all the neurotransmitters that affect the organs of the peripheral autonomous nervous system act indirectly. Those are the indirect activations. Norepinephrine, acetylcholine and serotonin, another central nervous system neurotransmitter, all produce state changes in the central nervous system. One important example is the change from the sleep state to the wake state. That is one thing, and that needs to be done slowly. That is known as a wakefulness or arousal. It needs to happen slowly, not that rapidly or fast. On the other hand, theories of learning and memory often invoke the action of indirect neurotransmitter to explain the changes in synaptic activity. These indirect neurotransmitters can modulate neurons and ultimately changing their channel function and producing a new and different types of output. And that is important for learning and for a long-term memory and the activities associated with memorizing something. So it's not a rapid task. It will build up slowly but steadily. So that in a sense are the things that we want to talk about. So let's look at the summary of what we've learned. We've learned so far that the neurotransmitter is released from vesicles in the presynaptic cell and binds to the receptor of the postsynaptic cell. Neurotransmission ends when the neurotransmitter dissociates from its receptor. The effect of neurotransmitters on the postsynaptic cell depends on the receptor, not the neurotransmitter molecule. If we vary the receptor, the response will also vary. Sometimes the response is inhibitory, sometimes it is excitatory. Acetylcholine and norepinephrine are the most important neurotransmitters in the peripheral nervous system, while glutamate, GABA and glycine are the most important neurotransmitters in the central nervous system. Glutamate work as a excitatory neurotransmitter in CNS, while GABA and glycine work as an inhibitory neurotransmitter in the CNS. And fast and slow synaptic activities serve different functions. Fast activity is required for the athletic performances. It is also required for the communication skills and other higher functions and sensory motor coordination, while the slow activity required for the learning and memory. That in a sense is the gist about this topic. I hope you understand the video. If you like this video, please hit the like button, subscribe to my channel to get more and more videos like that, and share this video with your friends. Thank you.